Good morning. Let's take a look at the exercise 3, which is the um, compound light microscope. So the objectives, you have these in your things to know information. Your objectives are going to be, you need to know how to carry and store the microscope. You're going to need to find the different structures on the microscope. We've got a handout there that we're going to follow and label all of these items. We're going to calculate the magnification. You're also going to need to be able to, for instance, if the microscope is sitting on the table, you should be able to look at that microscope where the objective lens is that you're being asked about and calculate the magnification. So we're going to go through that and we should be able to answer all of these questions. So I'm not going to test you about why it's called a compound light microscope, but bottom line is it's compound because there's two magnifications. There's the objective lens magnification, which these right here are the objective lenses. And then these are the ocular lenses, which also provide magnification. Keep in mind, ocular means eye. So it would make sense that that would be what you would be looking through. So that's why it's called compound. And you've got a light source right here, which is why it's a compound light microscope. Okay, so you do need to know how to carry and store the microscope. So you have got a labeling handout that you should have received in class. If not, there is one available on the Moodle page, but this is what that handout looks like. We're going to be starting over here on page 1, and we're going to be working all the way through to the end. So it's a 2-page handout. Put that back over there. So we're on page 1 of the labeling handout. It should say page 1 at the bottom of the page. And the first thing we're going to take a look at is how to carry the microscope. So you do want to use 2 hands to carry the microscope. You're going to have one hand on the arm of the microscope and the other hand on the base of the microscope. You're going to make sure that the cord is wrapped around and not dangling on the bottom. Now, you may be in a class where you have to retrieve the microscope or where you have to go put it back. So knowing how to carry it correctly is important. So on your test, what I'm going to ask you is I'm going to show you some pictures and ask you to pick the picture in which the person is either A, carrying the microscope correctly, or which of the pictures is showing a person carrying the microscope incorrectly. So I could ask that either way. So in this situation, in this picture here, which one is correct? That would either be figure A is correct or figure B is correct. So you should pick A. Now on your test, there's going to be more than just 2 pictures to choose from, but this just kind of gives you an idea how that question is going to look on your test. Remember, your test on this one, you're getting up, you're walking around the room, you're going from station to station. So at this particular station, there would be a picture in front of you with 4 images of a person carrying a microscope. And the question would read, which of these is the correct way to carry a microscope? And you would pick the image that would be the person carrying it correctly, or which is the incorrect way, and you'd pick the one that's carrying it incorrectly. So that's how that would look on the test. So it'd be an ABCD answer for that particular type of question. Now, when you store the microscope, you want to make sure, bottom line is, let me come back up to this picture here. Like, this is stored incorrectly, because if you notice that objective lens is right there next to the stage, you want to make sure that that objective lens is very high up and the stage is very high down or very low down, because you want to keep those glass pieces as far away from each other as possible so there's no chance of them getting broken. So that's really the big idea. So you want to make sure there's no slide on the microscope anymore. You want to put the smallest objective lens in position, which is the scanning one. That's the shortest one. Lower the stage, coil the cord, make sure the power is turned off, and you unplug it, obviously, if you're going to go put it away. Now, what are the parts of the microscope? So you should be on page 2 of the labeling handout. Page 2 of the labeling handout is going to look like this. So you should be on this page now. And we're just going to go around the document kind of clockwise. So we're going to start at A, then we're going to go to B, then we're going to go to C, then we're going to go to D. And your PowerPoint that I'm going through right now does that same order. Okay. So as you can see here, I'm just kind of revealing the letter A. Okay, so letter A, this area right here is the head of the microscope. It's where the objective lenses are attached. Letter B, you can see right there, letter B is the arm. And remember, this is where one of your hands is going to be when carrying it. Letter C is pointing to this outer ring, not the inner ring, but the outer ring. That is the coarse focus knob. And what that does is going to make big adjustments. So that's going to allow the stage to make big adjustments upward or downward. I'm going to change colors here just because I don't want to mess that up. So with the coarse adjustment, coarse focus knob, that's going to cause the stage to visibly go up or down. Now, letter D is just this little inside circle, and you can turn that, and that's a fine focus knob, and that's going to make minor, very small adjustments to the movement of the stage up or down, to where if you're looking at the stage, you're not really going to be able to see the stage moving at all. But if you look through the microscope, you'll be able to see things come into focus or out of focus. Now, letter E is the base. And I put on here, it's not going to be asked on the test, but I did put on here where the light switch is, the on-off switch, and the light intensity knob. So this is like your dimmer switch, okay? So you do want to make sure that when you turn the microscope on, that there's light coming from in here. Otherwise, the light bulb could be burned out, or it could be not plugged in, or maybe the dimmer switch is in the dim position. That's the light intensity knob. So you want to check those things to make sure. Letter F is the sub-stage light. There's a little light bulb in that spot right there to provide light to go up through the specimen. Letter G, now this is pointing to something that's really, really small, so you kind of have to look under the stage to see it. It's called the iris diaphragm, and it's an aperture that opens and closes, and it doesn't close all the way, but it has either a pinpoint availability for light to pass through or a larger diameter circle for a lot of light to come through. So it's kind of like the iris of your eye, okay? Letter H is the stage. Now, this stage also has these knobs right here, which we're not getting into, but they are going to allow the stage to move right and left and back and forth. So the coarse and fine focus knob allow the stage to move up and down, whereas this moves the stage. This is the kind of microscope we have. It's going to move the stage backwards, forwards, and right and left. So it's pretty cool. We don't have to move the specimen around to be able to see things. We can move the stage around. And then letter I is pointing to these structures right here. These are all magnifications. So these are your objective lenses. And there's 4 different objective lenses that we can see here. I only have arrows pointing to 3 of them, but there's one right back here. And they all magnify the image differently. So there's one that's magnifying it by 4 times. There's one that's magnifying it by 10, one that's magnifying it by 40, and then the other one would be magnifying it by 100. We are not going to be using the 100 power objective. And then letter J is the revolving nose piece. So this rotates to put a different objective in position. Now, actually, the objective that is going straight up and down, the one that's vertical, is the one that you're looking through. So the ones that are kind of coming out here at angles, you're not actually looking through those. So the way this microscope is positioned here, the objective lens that is going straight up and down is actually the one that's being used. And then letter K, these are the ocular lenses. And because this is a compound light microscope, we said there are 2 magnification locations. This also has a magnification power of 10x. So it magnifies the image 10 times. If you look on the ocular lenses, it tells you it's magnifying it 10 times. So it's not a secret. It's right there on the lens. So that's those are the structures on the microscope that you're going to need to identify. So how am I going to ask the question on the test? Well, there's going to be a microscope sitting in front of you. It's not going to be on a sheet of paper. It's going to be an actual microscope. And I will put a sticker right here and say, name that structure. It's supposed to be a question mark, by the way. Name that structure. And then on your answer sheet, you would put arm. If I put a sticker on this piece right here and put a little question mark and said, name that structure, you would say stage. So you're going to have to look for the stickers on the microscope and then you would answer accordingly. So that's how the question is going to be on the test. Reminding you all, because this is our first test, that there is no word bank. So you're going to have to come up with these names on your own. So how would I study for this? Well, maybe I should have told you this before we actually filled out the labeling handout. I would take your labeling handout and maybe on a separate sheet of paper, write down the letters A through K and write the answers down. So that way, you know, A is the um, uh, head of the microscope, B is the arm of the, oops, sorry, B is the arm of the microscope. So that way you know what the right answers are. And then you have this blank one, and you could do like A, B, C, D, all the way through K on a separate sheet of paper, and then practice that. Okay, what was A again? What was B again? What was C again? So write it down over and over again, but you've got a clean sheet of paper to work with. Again, this is also on your Moodle page. Okay, so you can have a blank copy on your Moodle page as well. So let's take a look at page 3 of the labeling handout. Now, I will say this is no longer on page 3. This is actually on page 2 still, so you don't have to turn the page. I condensed it to just 2 pages. So what we're going to look at is calculating the total magnification. So we said there were 2 magnifying areas for this particular microscope, the ocular lens and the objective lens. So how do you figure out what the total magnification is of the object that you're looking at? Well, the objective lenses have magnifications written on them. So right here, that's 4 times the magnification power. This one is 10 times the magnification power. This one is 40, and this one is 100. And if you notice, the length of the objective lens gets larger as the magnification increases. They're also color coded, so there's a color scheme going with them as well. This is not a secret. So look at the microscope that you're being shown on the test and just look at the number on the objective lens. You don't have to memorize it. You do need to know, though, it doesn't have the name. So in other words, the lens that magnifies it 4 times is called the scanning lens. So that you need to know. The 10x magnification, this one right here, the one that has the yellow band around it, that is the low power objective. The blue one, the one that has the blue band, is the high dry objective. And then the white one is going to be the oil immersion lens. And you can see it right here. That's the oil immersion lens right there. We are not going to be using the 100x magnification. So your microscopes will only have 3 objective lenses in place. The 100x will not be present. So when you're calculating total magnification, this is just going to be either a word question or it's going to be look at the microscope as it's sitting on the table in front of you and calculate the total magnification based on the alignment of the microscope. So I'm going to show you kind of how we're going to do this. So if I said if the scanning lens was in place, now you should know the scanning lens is 4 times the magnification. Remember we said the ocular lens was 10 times the magnification. So what you're going to do is just multiply the 2 numbers together. 4 times 10 is going to be 40. So if the scanning lens is in place and you look through the microscope, you're going to be looking at an object that's magnified 40 times its normal size. Now, if the low power objective lens is in place, that we said was 10 times the magnification. You're going to multiply that by the ocular lens, which is 10. So the total magnification when looking through the low power lens is 100. So that's all you would put on your test, just 100. Now, if the high dry lens is in place, that is 40 times the magnification multiplied by the ocular lens, which again is 10. These numbers are on the microscope, so you don't have to remember them. You just have to remember high dry is 40. This would be 400. So if it's on the high dry lens, you're magnifying that image 400 times. And even though we don't have the oil immersion lens, we'll go ahead and do the math on that. The oil immersion lens is magnifying the image 100 times. Multiply that by the ocular lens power for these microscopes, which is 10, and you're getting an image that is magnified 1,000 times. You're going to be looking at the oil immersion lens when you're in the microbiology class. Okay, so in this image, sorry, it's a little blurry, but if you were looking at this and this were your test microscope, what you would do is you would look at the objective that is straight up and down. This one is the 10x objective. You can see it right there. So if I was asking you for the total magnification as this microscope is sitting, you would take the 10 for the objective lens and 10 way up top for the ocular lens. So that would be 10 times 10. Your answer for the test would be 100. That's what you would put on the answer sheet. No, you don't need a calculator for this. You can just add a 0. If it were this situation here, so again, what's the blue one? It's going to be 40. Again, you can't see it in this, in the image here, but if you just kind of tilt your head to one side, or, as you're looking at the microscope in front of you, you can kind of move a little bit to see what the number is. But in this image, this is the one that's straight up and down. So we would take 40 for the objective lens, multiply it by 10, which is the ocular lens, the one you look through with your eyes, and that would be 400. So the way this microscope is sitting at the moment, if you were to look through it, you would be magnifying the image 400 times. And that's it for the microscope.